Hello, everyone. Welcome at the first round of creativity pitches. Um, First, we want to show you the 1 minute pitch of Twente because their pitch wasn't played at the opening. So, first, we are going to look at that.
More presentation this side? Because it wasn't coming indeed. More presentation this side?
No, this is only last meeting for the max
Sound
volume,
for
right?
the rest of the presentation.
This is the max volume it goes. Can't get louder than this.
No, because we have to make sure. Now we have to change the setting for the sound. It's not that easy.
Start the video, make it louder. It's okay if it's not on the pitch.
But it's,
Play it again.
I don't think it gets any louder.
No, but we have to make sure. It's a little bit complicated because we have to put extra wires from our mixer system to this and back again. So
But you know if this gets louder in the room?
no.
And then let's just continue.
Just say sorry.
This is the only one. Sound?
Yes. Toch? Yeah. Okay. Sorry, the sound wasn't getting harder, and I'm sorry for that. But we will continue with the pitches. Every team, there are some persons or someone who is presenting, and the other students from that team who might answer questions. They can sit here in front at these chairs. So we're gonna start with the first pitch, which is from True Sense.
Thank you. Might I start it? Good morning, everyone. I'm Chen Chen from True Sense China, and today I'm glad to be here to share our creativity with you. First is our design working principle. We use an optical strategy to build our biosensor, which is composed of 3 parts of spectrometer, our frame, and our microfluidic platform. Our microfluidic platform was composed of 2 layers. For the upper layer of a microfluidic channel and the lower layer of a chip design. And the 2 layers were put together by a clamp. And this is our principle. Once you apply the blood sample in the inlet and it flows through the microfluidic channel, and the biomarker of GFAP will be captured by the antibody on our substrate, and the absorbance is thus shifted, and which can be detected by our spectrometer in the lower side. And let's move on to more details about our design. First is the selection and production of our affinity element. We choose it as a nanobody. Why we choose nanobody is because it has a smaller molecular size and it can interact with the ridge and the groove area of the antigen site. And we utilize a phage display strategy to produce our nanobody. And the reason why we choose phage display is because compared with traditional ways, for example, the hybridoma or some bioreactors. The phase display is easy to modify, and the selection cycle is rather short, so we can do a lot of so make us able to do a lot of modifications. And our current corresponding progress is attached here. And now we move to our chip design. The microfluidic channel, as we can remember, is composed of two layers and clamped together. And we designed it to slow. We simply designed it to slow the rate, slow the flow rate of the sample, so it can evenly distribute over the chip. And we did some microfluidic simulations, as you can see from the picture in the right corner. The simulation shows that the flow indeed is very even on the substrate, so it shows our design really work. And to make our design more feasible, we did some perspectives on the microfluidic channel. For example, we can add a blood fluctuation to fill out the blood cells, or do some pre-treatment, for example, add a blocker in the substrate and can eliminate the bifolding. Also, we can add an eluent inflow to increase the usability of the chip, so it can be recycled after use. And currently, our design was use a spectrometer, which was costly and detects the whole spectrum. To cut the cost, we would perspective outlook to use a CCD camera to limit the whole spectrum to certain wave band, so we can directly detect the light intensity instead of detecting the whole spectrum. And that's all. Thank you for your comments.
Okay, thank you. Are there maybe questions?
Because GFAP is a very long protein, so it has many ridges and grooves in the protein and the configuration. So we use a smaller weight nanobody, so it can easily interact with the ridge and the groove, like this one. I'm sorry. But can get me. Okay, thank you.
Any more questions? Maybe from the jury? Oh, yes.
Label. Yeah. We want to limit the wave band to 500 to 550, maybe more accurately, but in this wave band, like label, we don't have label. It's a labelless detection technique. What, can you say it again?
You're using antibodies to detect the compound, so this is single use, right?
Yes.
Yes, so that's why we choose nanobody, because nano antibody is cheap to produce, so even it's single use, but we can create it through the bacteria. That's really easy.
For the detection, it takes only about 1 minute, but for the bio, we may take around 5. For the bio.
It depends on the fluidic speed, but we think it's really fast because we have decided for about 5 minutes, and actually the full edit only takes 1 to 3 minutes.
There was a question from the jury.
Based on your report, my question would be, so you also showed here that you do electrochemical measurement at the same time, right? So is it, your system is really suitable for both optical and electrochemical measurement? What kind of changes do you do to your mass spectrometry so that you can or you use exactly the same system?
There's actually two separate systems, and the electrochemical is our back plan.
So why you decide optical then?
Because we want to add some more creativity to our traditional electrochemical one, and want to do some innovations.
Exactly, the electrochemical plan is our backup plan.
Okay, maybe. Yeah, I think it's okay. Just to follow up on that, then, did you do an investigation into whether the optical then performed better than your backup plan? Do you have any thoughts on the comparison between the two techniques?
Exactly, we want to try something new because the past years we tried the electrochemical methods and we use NIP. So this year we want to try some new and try the optical measurements.
Right. And did you feel it, did it work better than the old electrochemicals? Or? Sorry. Oh, yes. No, I'm asking if then, so you use the optical method this year, but did you have any experience with whether you got better results with this technique compared to the backup?
In fact, I think we need more development, and so that's why we have our future plan on the maybe page 4 or 5. That's because it takes only 1 year to do these optical experiments. It's too short for us, so we need to try something more, make it more accurate. But it seems that to us, the backup plan is more stable.
So, if you would put it in 1 or 2 sentences, what is the most creative part of your sensor? Where's the strongest creativity created?
In fact, we divide it into 2 parts. One is for the optical measurements, and one is for the biology. And that's why we have a very special department for biology, nano antibody design. But I think, as you can see, we are 3 major students here, and maybe in my angle, I think my phage display is the best creative part. But as what I know, my friend didn't know. My friend didn't think of that. He thinks his microfluidic is the best one.
Last question.
I had multiple, but I guess I will ask only this question then. You use for the detector a CCD camera, and you are measuring particles. Are you using the average signal overall, or are you using the signal from each pixel?
Well, actually, as you can see, we use the spectrometer for now. And as the CCD camera, we can detect maybe some wavelengths. We can detect the wavelengths, some wavelengths. It's promotion, I think. That's where we are working now. And for now, we're just using the spectrometer and detect the whole wavelengths.
The sum up of the 510 to the 530?
Yeah, 10 to 40, yeah. Mainly between the 510 and maybe 550.
The sum of the narrow spectrum during this part.
And we can make it narrow.
Thanks.
Yes. Thank you for your pitch. Okay, it's time for the next page. And that page is from BioSensum. I think here you can see the time. Good luck.
Good morning, everyone. So thank you all for being here. We're from Montreal, and today we'd like to present GFET sensor with using a portable readout system that is quite optimized to the task. But first of all, just a recap on GFAP. You're going to hear about it all day. Basically, it's a biomarker for brain injuries, and the usual patients of these injuries need to know it within minutes because it usually occurs during sporting events, and we don't have the time to bring them back to the hospital, and we need on-site devices that are optimized and customizable and efficient to use. So, briefly, the field effect transistor made of graphene, typically you have a source and a drain channel, and its conductivity change depending on its environment. So there's a neutrality point in which we can observe, and we will exploit that. It's basically a transfer curve. It's the direct point. And also, as you can see on our chip design, we use multiple channels. The purpose of the channels is to increase the accuracy because for example, 16 channel, we can do an average and have a better signal. And on figure 2, you can see the graphene, the source drain, and the interaction that we will see further. Surface chemistry is quite simple. You have a linker for the graphene. You have the antibody to capture the analyte. It's quite simple. And the transduction goes as follows. So you can see the shift in the environment. So if you have bare graphene, the neutrality point occurs at around around 7 millivolts. And as you add more stuff on the environment, the shift goes to the right. And it's the same with concentrations of analytes. For the introduction of the analyte, we have a simple microfluidic system. It's basically stropwise. You drop the analyte 20 microliters on the tip, and it spreads on the graphene. Now the good part. So the readout system, it's fully Arduino powered. Why did I want to do that? We wanted to do that. It's because with semiconductors, there's a lot of variability. With the surface chemistry, there's a lot of variability, and you need different accuracies at different points for different tests. So the topology is quite simple. You have on the first PCB, you have 2 voltage generation patterns. So it's basically a DAC followed by a buffer and an RC filter. One is for the gate electrode, and the other is demuxed into 16 channels, right? And what makes this versatile is the green part. So you have the little resistors. You use a voltage divider topology,
to reinvent the wheel. And so you have 16 resistors in our case, but they're integrated on a removable chip. So if I want more or less, for example, if I want detection down to 1 milliamp from the transfer curve I showed you earlier, then I put 1K resistors. If I want to go up, I put 3K resistors. But there's also another problem, which is if you go too precise, then you cannot, there's a set amount of current introduced to the load resistor, so you have to optimize. And this is where the tunable non-inverting amplifier comes in. You can, when you amplify back the voltage accompanied with a PGA, since we're working with Arduinos, we can go crazy. We can bring so much stuff on a baseball field. So this combination here can transfer the small signals measured on a GFET through a portable Arduino. Everyone can buy one, Amazon. It's cheap, you can have fun with it, but you can also make biosensors. So the whole thing is customizable. And for calibration, as I mentioned, you have different ranges of precision. There's a digital feedback mechanism, so you can establish a current curve. I do not have it on this PowerPoint, but basically it self-corrects. And as for the external calibrations, if you don't know what semiconductor and surface chemistry you're going to have, you can simulate one with pre-built chips with series of resistors that gives you, that simulates a transfer curve, right? There's the programmable gain amplifier, tunable, and the resistor chips are used for calibration. So that brings us back. You want a 9-volt powered battery device. Oh, I'm sorry. So having a fully autonomous, customizable, post-production, you can customize it post-production for different needs and different tests. It self-calibrates. It's powered by the 9-volt batteries. It consumes very low current. And you can change the type of test. It's good for electrochemistry. It's good for optical sensing. For example, if we use MOSFETs instead of GFET, you could combine with a different spectrometer, or you don't even need one. And it's modular. So the thing with Arduinos is that they have shields. So you can have one shield. You basically plug it on top of the Arduino, and it has already the integrated circuit on it. So if you want to do one type of test, one type of antigen, you can just switch it up. The system stays the same. Topology stays the same. And you don't need to bring your lab equipment on the field. Thank you for listening.
Yes, thank you for your presentation. Other questions?
Yes. Oh. P-base. P-base.
So yeah, P-base is a molecular linker with the carbon circle, so it can do a non-covalent bond with the graphene by pi-pi stacking. And the other end is a covalent bond with the antibody. It's really easy to use. It takes 10 minutes and it's on the graphene, so that is why we chose it.
Oh.
Yeah, obviously it's reduced the. Do you want to?
Sure.
Yeah, so basically that can potentially reduce sensitivity. And if we had a little more time, something we would have liked to explore is the use of SCFVs, for example, instead of full antibodies. And then I'm not sure if you're familiar with IMAC protein purification. So one thing we could do with that is leave the his tags onto engineered antibodies and attach them to NTA that is itself attached to P-base in the same way that you would purify proteins, and that would ensure better orientation of the antibody at the surface of the sensor. Yeah, so that's definitely an issue. Yeah, we haven't really developed a filter solution in order to have just a decreased salinity at the surface of the sensor. But yeah, I don't know if you want that.
But actually, we do the 5-minute incubation with the plasma, but after we do our measurement in PBS, so we wash it before we do the measurement. No, we do it in PBS, so we chose the ion concentration during the measurement.
I'm a little bit curious about the development of the electronics for sensing. So you showed like some filters and some IDCs. I would like to know how was like the step procedures to reach that circuitry as like our final answer, and how did you like explore the change in the transistor based on what you want to sense and what you don't want to sense, because if anything else binds, you also get a difference. So I'd like to know a little bit more about it.
First of all, disclaimer, I'm a chemist. So basically, we had the lab equipment, and the goal was essentially, I just wanted to try and reproduce what was seen on the calibration. So I had the bare graphene. I had the same 3 curves I showed earlier, right? Oh, sorry. Surprise. So basically, the gate voltage is no problem, right? But the thing is, the upper bound and the lower bound of the current input, I had to vary because actually of noise, like you mentioned. There's other interactions, but to be honest, I just tried different sets of voltages and resistors until I could get curves that matches. So if there were noises, it was already taken into account, because as I mentioned as well, we use statistical curves. Like, we use 16 channels, so the average kind of, it doesn't cancel it out, but it helps. Yeah, just replicating what larger instruments can do and small. Yes.
Question from the jury.
Yeah. How do you establish your calibration curve? Is this based on shift in voltage or ratio between the potential and the voltage?
So the calibration, so for example, let's say those are not the values. This would be concentration A and B. So green is A, green and B. Instead of comparing B to A, we all compare them to blue. So we have the bare graphene, we would have a set of chips with the antibodies and in the matrix, and and then that would be our calibration, and we do a set. We didn't manage to make a proper calibration curve with known concentrations, but it would be a typical standard. I don't think that answers your question.
Okay. So yeah, we look at the voltage of the VCNP. So yeah, that's it.
Then there's the last question.
Thanks. So as I understand it, you make a calibration based on varying concentrations of the analytes in the same plasma. So if you were to get a plasma sample from a different patient who has a different plasma composition, how would you reference for that?
I think it's about the selectivity of the antibody. We just want to detect the GFAP. So when we do our measurement, like I said, we wash it before. We wash the plasma and do it in PBS with the known ion concentration. So we would use blockers to block the graphene, but we didn't have time for this in this year. But do you have something else?
Yeah, so basically, since we're at the moment, we don't have the filter system, but the measurements are taken in PBS, so you would wash out the rest of the plasma before you take the electrical measurements. So that would allow your antigen to bind to the antibody probes. And then what, in theory, we would like to just keep like the GFAP basically at the surface, and that's when we can measure the different concentrations. But just to go back to the point earlier, so basically we take calibration, we take transfer curves after each step of assembly, so bare graphene following P-base, following antibody, to get characteristics of our chips. And then basically the first curve we take after the antibody immobilization is our initial curve. And then this answers maybe more your question before, but and then we take different transfer curves at different concentrations of GFAP, and we plot the shift between each curve of each concentration to get our calibration curves.
So that would mean that if you get a sample from a different patient, you wash away the excess plasma proteins, but any non-specifically adsorbed plasma proteins are part of your GFAP signal.
Yeah, so that's the thing, is we want to also work on passivating our graphene, our P-base, and any antibodies that haven't bound to GFAP.
So what is your strategy for that?
So for graphene, I think we can use BSA or Triton. Isn't that for P-base? Okay, and PBA for P-base. Yeah.
Thanks.
Yes. Thank you for your pitch. Okay, it's time for the next team. Swiss Sense. Oh, SensUs.
Did you know that almost 90% of Can I do it again? Okay. Okay. Did you know that almost 90% of minor traumatic brain injury result in developing conditions like Parkinson's disease and neurodegenerative disorders? Well, this unfortunate fact Can I go? So, did you know that almost 90% of minor traumatic brain injuries remain undetected? This unfortunate fact results in developing conditions like Parkinson's disease and other neurodegenerative disorders. Our purpose today is to present you an innovation that will change this situation. So, we developed a biosensor focusing on 4 critical aspects. First and foremost, we tackle the challenge of molecular recognition in order to bind the protein. Second, microfluidics, to efficiently flow our sample. Third, detection, to transform the protein concentration into a readable signal. And fourth, user experience, to reduce stress on medical personnel. Of course, we know that a precise and accurate diagnostics is important and hinges on the ability to identify and bind the protein of interest. This is why we've developed a sophisticated molecular recognition system. Our system involves a sandwich assay methodology with the mobilization of the antibodies on gold surface with oligonucleotides. To amplify sensitivity, we decided to integrate the antibodies on a functionalized gold nanoparticles. This safe, reliable, and robust technique allows us to detect concentrations up to the picomolar range of the protein of interest.
Efficient flowing of samples is essential in creating a robust and low-risk biosensor. That's why we created our very own microfluidic chips made entirely out of glass. After prototyping in transparent resin, our partners at Femtoprint created this design for us, our design, with femtolaser technology. The resulting chips boast over 90% optical transmittance in the visible range in near infrared, and the patterns in the glass allow us to use microfluidic inserts and O-rings to completely seal our sample. Next, the detection mechanism itself. So our system is an optical one, and we actually use commercial DVDs. When you cut a DVD and split it open, on the inside you'll find a metal reflective surface. That reflective surface will have a grating, and that grating will have a period of around 740 nanometers. This grating makes plasmonic coupling very straightforward. We use a broadband LED and collimate it. We shine it through the glass chip, through the solution, and it gets reflective off the metal surface. The resulting spectra shows a resonance dip that shifts its position depending on analyte binding. The only necessary modification to the DVDs is just adding a thin film for surface chemistry if necessary. In our case, we used gold. So we have a very simple to produce and cheap detection surface. Finally, user experience. We all know that user experience is critical in the adoption of any medical technology. That's why we wanted to make our interface easy to use, and at the same time still offering all the necessary functionalities necessary for scientific experimentation. We have integrated signal processing, and for the microfluidics, we have 3 modes of operation: manual, semi-automatic, and automatic, allowing anyone to perform an experiment. We're Team SensUs. Thank you for your attention.
Yes, thank you. Other questions?
Thanks, guys, for the pitch. So going back to your molecular recognition design in that slide, I had a couple questions. One, why did you select 2 different antibodies, and how does the sandwich assay affect GFAP? Does it improve hERT? And the second is, I think I saw in there, so capture antibodies with oligonucleotide. Could you explain what that means as well?
Okay, in order to select the antibodies, we did many experiments with a machine called biolayer interferometry. This machine actually, the purpose of the machine is like to give the shift of the shift in nanometers. And so I did the different combinations of the antibodies, of the detector and the capture antibody, and I found out that 19 and 15 were the most consistent, and they had like the most signal.
And so what was capture antibodies with oligonucleotides?
Yeah.
We used oligonucleotides, which we conjugated via amine binding to the antibodies. And these oligonucleotides consisted of a long strand of adenosine and then tenosin. And the adenosine binds electrostatically to the gold surface, and the tenosin then shoots it up away. So it's like an L shape, and then the antibodies sit right on top. And this has a couple of reasons why we use this, which we hope to explore further. One of them is specifically that they're quite strong and would allow for regeneration methods.
Hi, I still can't really make the connection between the slide on the molecular recognition and the DVD. So what is mentioned here as gold chip, is that then the surface of the DVD?
Yes. So we just needed, for a surface chemistry, we needed a gold surface. That's what the biology team gave us as a task. And then we could explore whatever optical sensing method we wanted, as long as we gave them a gold surface. And so these aluminum DVDs have a grating, and it's, as I said earlier, it's super easy to then shine a broadband LED, and we see a spectrum, and there's a strong resonance dip around 660 nanometers in our case.
And that shifts due to the gold or due to the patterning on the DVD? Because I mean, it could also be just an optical resonance coming from the pattern itself.
So the pattern allows for the plasmonic coupling, but then the plasmons propagate through the gold film.
Okay, so the spectrum basically shifts, yeah?
Yes. Yes.
Okay, thank you.
And so it's, of course, it's refractive index based, as all SPR machines are. We wanted to go for a super simple, super easy to produce system that ideally anyone in a high school setting or whatever could maybe try to replicate. We thought that was a fun idea.
Thank you.
So thanks. So super simple, super easy to use. That's nice. So where did this idea come from? So where was the creativity in your team?
You mean in general, the creativity in the team?
The idea for the concept.
For the DVDs?
How did you create that?
So initially, we were brainstorming, and we wanted to go for a smartphone-based portable system for ambulances. And so we were thinking, how can we make it really cheap, small, and using basically stuff that you already find on the market, and not only ambulances, but also maybe at home testing, and et cetera, et cetera. Since then, we did market research, and we realized that wasn't a viable business plan, and so we completely switched up. But that's where it comes from. We wanted something where we just use a smartphone camera and a DVD chip, cheap, easy. That's what we were going for. But we kept the DVDs because we thought it was cool.
Okay, good.
There's a question.
Do you measure in serum, PBS, or molecular water the moment of measurement?
That's maybe Maron.
I want to.
Okay. So basically, the final measurement is after a wash of PBS. So we first flow in the serum, then there is binding, and then we do a cleaning by PBS, and then we do the measurement.
Is there one last question still? Okay.
I would like to know a little bit your experience with the use of the broadband laser. You mentioned like the coagulation and like like select the laser that you want to use, how to try the coordination, and also the user interface. Where is it developed? Is it on a onboard chip? Is it on a notebook? How can you tell us a little bit more about it?
So the user interface is on a laptop next to it. We wanted to integrate it on a touchscreen at first, but we didn't really see the points of it. Again, it's all coupled with the market research, and we realized if it's in a clinic, if it's in a setting for, I mean, you'll have to listen to the business pitch, but it just didn't make too much sense, and we also, honestly, we didn't have much time to integrate it. The first question was?
The laser, the coordination of the laser.
Okay, yeah.
Choose the laser itself.
So it's we were playing around with different coatings on the DVDs. We were playing around with the orientation of the gratings, because if you oriented it in a certain way, then you polarize the light in 190 degrees versus another. If you turn the chip, then you have the gratings at 90 degrees, and you need to polarize the other 90 degrees, and you see the shift is quite different. And so we were looking at shifts, we were looking at dips around 500 to even 850. So we wanted something that could give us light invisible and near infrared. And so we used a broadband LED that does exactly that. It gives us relatively consistent light from 400 to 1050 nanometers. And then collimation, all the optical setup, we actually, as you can imagine with one of these projects, the first prototype is absolutely terrible. And we first did it in plastic and wood, and we were like, okay, this is impossible to align. We were using Scotch tape to try to align everything. But so we switched everything to metal, and now we have everything in aluminum, and it works much, much better. It's super easy to align now. But yeah, there you go. Hope that answers.
Okay, thank you, SensUs. Okay. It's time for the next team, and that is Twenties. Make sure to use the microphone.
Yeah. Thanks.
If you stand there, you won't be in front of the PowerPoint.
Dear all, welcome to our presentation. My name is Diana Andreoli, and as the team captain of Twindos, I would like to introduce you our TBI biosensor platform. Traumatic brain injury is a damage to the brain that causes lasting consequences. This injury, while this injury happens, depending on the intensity of the injury, GFAP is released into the bloodstream, and this can be used for the assessment of the severity of TBI. This is crucial for the assessment of mild TBIs, because these mild TBIs are often omitted or not diagnosed, and a lot of these cases also happen in sports. Our proposed solution is a user-friendly portable biosensor platform that is able to swiftly and accurately detect mild TBIs using electrochemical impedance spectroscopy, which is, in our case, label-free sensing, and it's a platform technology. For the surface functionalization, we make use of the electrochemical affinity of tiles to gold surfaces, and these tiles we further have biotinylated to be able to make use of streptavidin-biotin interaction. And after this step, we would like to attach the antibodies, but the antibodies first need to be biotinylated as well, and for that we make use of a technology called oil linkers that are photoactivated to be able to attach a biotin group to the antibody, and then after that we can attach the antibodies to our layer as well. Electrochemical impedance spectroscopy for this we make use of gold-plated electrodes. We have designed our own interdigitated electrodes that are grooved. We have made COMSOL simulations to optimize the sensitivity and to be able to have the highest sensitivity of the electrodes. For the electrodes, we make non-Faraday EIS model fitting because we measure the resistance of the bulk that is very close to the surface, and we make use of the Bode plots to be able to read out the results. After proof of concept of this setup, we wanted to shift to a faster testing to be able to make a proof of concept of our whole device setup. So we shifted to off-the-shelf planar electrodes that we could purchase. So for this technique, we have to shift to Faraday EIS model fitting. And to increase the sensitivity of this technique, we are also using redox mediators, which is the ferrocene in our case. For the cyber-physical system design, we make use of the Python clutch that encapsulates the cartridge with the chip. Further, our technology, our setup is including a Raspberry Pi that controls the whole system and MSTAT Pico to make the measurements. We have containers for the buffer, for waste, and this whole system includes also the micropumps, and everything is controlled by the Raspberry Pi. So after the sample of the plasma is inserted into the device and we start the measurement, everything is automated and all the pumps start running and the measurement is done, which we will after see on the results on the screen. For the design ideation, we have gone through several iterations of the device design for the optimal user use design. And for our alpha prototype, we have gone with the last design you can see on the right side for the proof of concept of our device. For the graphical user interface, further we have included multiple buttons on the screen. Mainly you have the run the test button to start the measurement, plus you will see the readout of the concentration of the measurement on the middle of the screen and the scale. They will show the severity, plus some extra buttons for extra information. I would like to still recap the originality of our device, and mainly want to give some attention to that our cartridges are recyclable and can be reused after they are shipped to us. We can clean them, reuse them, and this point-of-care device can be used in various applications, but mainly we are focusing on elite sports. And I want to also accent on the fact that it is scalable for a range of biomarkers. I would like to thank all of our collaborators and all of the support we have received. Twentas, keep your head in the game.
Thank you for your pitch.
Thank you.
Other questions again?
Why did you use EIS instead of optical methods? Such reason?
Electrochemical methods generally are downscalable. You can make them much smaller, cheaper to produce, and easier to run. There's less points of failure throughout, so it's with the outlook into the future.
You mentioned something that you could reuse the chips. What kind of regeneration buffer are you using for
that? It's called sodium borate, and that will remove the tiles from the gold, and with that, so the tile gold bond will then break, which will lift off the entire biolayer.
Secondly, we can also use stronger cleaning, which is like piranha-based cleaning, for example, or piranha acid cleaning, but this can damage the electrodes too, so they are not so long-lasting. There will be a certain number of times you can do this cycle to clean them.
Thank you for your pitch. I have first question. Can you explain the technical differences between your Faradaic and non-Faradaic? It seems to be based on the electrogeometry. If that's the case, why was one different or better?
Yeah, so we started with non-planar electrodes for high sensitivity, trying to reduce the dye volume extended the bite length, and so increased the sensitivity close to the surface. But the chips were hard to produce, and the yields from the clean room were a bit low. So, and the sensitivity was high enough for us to switch to a much simplified planar geometry available off the shelf. As for the Faradic methods, if you could go back to that slide. Yeah, so with the high enough sensitivity with a more complex grooved geometry, switching to a simplified geometry, the sensitivity fell, and we decided to switch to Faradic with redox mediator. And the reading out of the results from the Nyquist plot with varying charge transfer resistance as like calibration curve parameter was also much more optimal than use of boulder plots.
Also, I would like to highlight, so for the grooved electrodes, we have been able to make a proof of concept with this technique also is more sensitive and we don't need a second like a redox mediator or anything for that, so we would prefer this option, but the optimization of the fabrication of the electrodes takes some time. So after we have been able to prove our concept with these electrodes, we shifted to the planar electrodes to be able to continue our whole measurement and our whole of the setup to finalize our design and all of the measurements.
Cool. And the second question I'd ask is, you guys bring it up, this reusability or sustainability. So EIS is extremely surface sensitive, as you guys probably realized.
Yeah.
Did you have a scheme for, so the thiol gold bonds are not particularly thermodynamically stable over long periods of time, and you talked about this being a mobile device, so you would have to store these. Did you test, or did you have a scheme for assessing how they drift over time or reliability over time of the device?
So yeah, like you're saying, they're not super stable. So as of now, we have been storing them in the fridge to keep the stability as much as we can. We did not have enough resources to test them for a longer period of time to know how much it would, how long is the half-life of the device would be and of the cartridges. So this would still have to be optimized further with more electrodes and with more testing.
Yeah, thank you.
Thank you.
So oil linkers are a proprietary technology from a company where they have an 8 kilodalton oligonucleotide, which goes and binds to only the heavy chain of certain specific antibodies. So when we introduce this, you shine 365 nanometer light on it for about 2 hours, and it photolinks your either biotin or fluorescent labeling molecule or something like that to the heavy chain of an antibody. So it was pretty easy to use right now in the proof of concept, but further down the lane, we would like to have antibodies that already have a biotin-based tag on it. So we can already produce them in the molecular biology labs.
Yeah, well, so for the measurement to make sense, we first take it through the first washing cycle. For non-Faradic AIS, that would have been just PBS, but when switching, then ferricene also has to be added. And then after sample is added and incubated for a minute, a second wash cycle of the same substance is used.
So the device contains 2 micropops. One is actually for pulling the serum through the chip, and then there's another micropump that adds the ferro automatically into the buffer of the chip. So then it gets pulled again. That way the device is fully automatic. So you only have to add the serum.
So your measurement is in PBS
Yes.
eventually after.
Yes, we make a blank measurement at the start in PBS, and then we take a final measurement in PBS again after the buffer. Ferrocene, I mean, yeah.
Do you think, because in all, any electrochemical sensor, you have the wide-length limitation, and your sensor in bioassay, you have 10 nanometer of antibody, something's on the surface, so how would you overcome that? I mean, because in the PBS you will not have that much of, I mean, you will have a wide-length limitation of around 2-3 nanometer, not more.
Just about the layering.
Well,
yeah,
I think for the non-ferritic EIS, that was our limitation, and we were trying to combat that by choosing the resistive regime of operation. But then, of course, switching to ferritic EIS, the problems still persist, but the sensitivity is, by the looks of it, enough for the biolayer that we've built. I hope that answers the question, not in full. But.
So we are adding the ferrocene to measure the resistance of the charge transfer. And yeah, there should be enough to measure that.
Okay, thank you. Unfortunately, we don't have any time for more questions. So thank you for your pitch.
Thank you.
Okay, it's time for the next team, and that's team test.
Dicht mijn mond houden? Oké, en de knikker bewerken. Ja, oké. Dankjewel.
Hello everyone, I'm Sam, and on behalf of Team Test, I will be pitching to you about the creativity and concept of our biosensor. Our sensor measures GFAP, which is a biomarker for traumatic brain injury. Even very low concentrations of this protein can already yield important information to medical professionals. So therefore, the main challenge lies in being able to detect such low concentrations. For this purpose, we designed NeuroTest. The main principle underlying our sensor is plasmon-enhanced fluorescence using gold nanorods. These rods are very small particles that have a characteristic plasmon resonance wavelength. This means that if you illuminate these particles with light of this wavelength, the electrons within the particle start oscillating, and this wave motion generates an enhanced electromagnetic field around the particle. If a fluorescent dye is present within this electromagnetic field, the intensity of its fluorescence is enhanced. And this principle we use to our advantage in being able to detect very low concentrations. We developed a chemical assay where a lot of these small nanorods are bound to a glass slide. They are also functionalized with capture antibodies. If we add blood to the sample, only GFAP proteins will bind to these antibodies, while the other proteins will just be washed away. After that, we also add a detection antibody, which also attaches to the GFAP. This is also attached to a fluorescent dye. In this way, the fluorescent dye is positioned in such a way that it falls within the enhanced electromagnetic field of the particles. This means that its intensity is enhanced when we excite the dye with a laser. This results in the end in an increased sensitivity of our sensor as compared to traditional sandwich fluorescence assays. We place our sample inside an optical setup that we designed ourselves specifically for this purpose. We direct a laser beam onto a dichroic mirror, which reflects the light onto the sample through an objective. We chose the wavelength of this laser so that it both excites the rods as well as the fluorescent dye on the sample. It's important to notice that the wavelength of the light emitted by the dye is higher than the wavelength of the laser. This means that we can essentially filter out the light that comes from the laser, so that in the end we only keep the signal that comes from the fluorescent dye. After passing through the filters, this reaches a camera, which forms a two-dimensional image of the intensities measured from the sample. Here you can see what such an image looks like. You can see a lot of small dots, which are all nanorods with varying levels of intensity. After applying image processing techniques, we can relate the intensities measured in this picture to the concentration of GFAP that was present on the slide in the first place. This results in a very good calibration curve. We managed to reach a limit of detection of 16 picograms per milliliter. And I would like to emphasize that this was acquired through measuring blood plasma. I'm very proud of the potential of the neurotest, and I would like to thank our sponsors who have helped us through their funding and sharing of knowledge in being able to develop this sensor. Thank you all for listening, and make sure to neurotest.
Thank you. I see there's a question.
I'd say that we made it so that there is little interference from background light. Indeed, vibrations would still have an impact because we have the sample. Small movements within the sample already result in a large difference within the of what you can see with the camera.
So it should be on a fixed table?
Ideally, yes. Yeah.
So I saw your setup was plastic. I mentioned before that we had huge issues aligning it. And you're using a beam splitter and two filters.
Yes.
And you still managed to align it that well to get the single light spot.
So the laser beam is indeed, it does reach the objective, and the objective, the main purpose of the objective is magnifying what we see on the slide, but it also helps in focusing the laser onto this single point.
In your setup, you did that?
Yes, it does.
You said that you wash away all the serum proteins, so I guess I have to ask you the same question as the other team. What about non-specifically adsorbed serum proteins?
We can see some results of non-specific binding on here because there is noise in this picture. You can see there's bright spots, which are the rods, but then there's also smudges on the picture. But the image processing techniques that we apply mostly get rid of this.
Yes, and we also use BSA to block the glass surface. So there's non-specific, to prevent the non-specific binding of the serum proteins.
Thanks.
So thank you for your pitch. The use of this nanorods, this is quite creative. So where does this idea come from, or how did you create that?
So as a team we started brainstorming and we came across this idea in literature, but it is also used by a research group here at the university that used like a similar principle.
So how long it takes all the measurement and analysis? From introduction of sample, washing, and analysis as well?
Yes, we first add the sample, and then we have now incubated for 10 minutes, and then after the barge we also incubate the dye for 10 minutes. But we have now used an open sticker, and we believe that if we use a flow cell or a microfluidic circuit, that we can optimize this to less than 10 minutes.
All processes 10 minutes. Altogether.
Yeah, and then there's the processing of the image, but that is quite fast.
Yeah, very good.
Okay, I'll first answer your question by explaining our thought process in choosing this method. We also considered other nanostructures, such as nanopyramids, but we found that the range where the enhancement occurred with these pyramids was very strictly bound to a specific range. In the end, we chose nanorods specifically because they have quite a wide range of where the fluorescence of a fluorophore is enhanced. And there's still variation in that because at some points the electromagnetic field is stronger than at other points. But because we measure a lot of nanorods at the same time, it averages out over a lot of them. And in image processing, we also take the average intensity over all of the nanorods that we can see. So that kind of averages out. No, we did not do such experiments ourselves. That we found through literature that this does yield more sensitivity than without using this enhancement. Yeah, but no, we did not experiment that ourselves.
Your sensitivity for the nanorods, it's, definitely on the edge is more compared to the other parts of the nanorod. And you also have another variation of nanorod sizes because definitely they are not all super monodispersed. So with all these many nanorods in your field of view, how did you assess and calibrate it so that, because based on these differences, you will get different signals to some extent. So how did you assess your sensor? Considering all the variations of the nanorod size and finding on different sides of nanorod, because on different sides you will get different signals.
I'm not sure if I understand your question correctly. How did we assess the performance of our sensor? Well, we measured the concentration range. Sorry?
The variations in the nanorod give you signal differences. The size and where the binding happens.
We in the end we went just settling for the idea that it averages out. So we did not have time to measure quantitatively these variations. And indeed, there are a lot of variables involved that can influence the intensity measured of one nanorod. So we assumed it averages out over when you just take a lot of nanorods.
There's time for one last question.
Okay, thank you. I think I asked the first team as well. So if you would say it in 2-3 sentences, what is the most creative content of what you developed? Where's the strongest creativity?
It is in applying the plasmon enhancement of gold nanorods in a point-of-care biosensing system.
Okay, thank you, Team Test.
Thank you all for listening.
Okay, the next theme is detectors. Make sure to use the microphone.
Yeah, I would.
Good luck.
Hello, everybody. Okay. So we are detectors. We represent the Technical University of Denmark. And I'm here to present our principle. So a little bit of why we actually are doing this. I myself had a concussion that still affects me today because it wasn't diagnosed early enough. So I'm still working through some migraines and everything, and I know I'm not the only one, which means this is very, very important. So the quicker we diagnose, the faster we can actually get the treatment started, and the faster we can actually start the treatment, so we actually can lessen the long-term effects, which will help a lot of lives, because of course the long-term effects are some of the ones that can really, really affect both the system and the person herself, and the lives around the ones that are affected. Yeah. So to do this, we started by setting up some criteria for what we wanted our biosensor to work, because one thing is knowing what we can do, but also what does it have to be able to do before we can actually figure out which is the best way to do it. So we wanted it to be easy to use in a hospital setting. We didn't want it to take up too much time, because the faster we can get it, the easier it is, and also we take away from the testing. So it has to be accessible, so it should be easy to have in the lab and in the hospitals. And of course it has to take low concentration, because different concentration of DFAB can lead to different treatment plans. So we were sitting down in our group saying, okay, what experience do we have? What have we worked with? Which different biosensors can we figure out that we can use? And ended up using electrochemistry, because some of the group had a bit of experience in it. So we kind of sat down and did design, okay, how do we do this? Okay, we have our electrode. We need it to immobilize the antibody. We had different antibody we tested to figure out which one was the most optimal, like how did we get the most specific result. And then we kept going back and forth and optimizing to be as precise as we could be within the time. So we are using electrochemistry. We have a small electrode where we immobilize the antibodies on it. So as you can see, this is very small. We during the testing of it, a lot of people came over and tried looking for a biosensor. They thought we had an empty table because it took up so little space, which was one of the ones we wanted to do, because we know already in a lot of labs and the hospitals, the space is already occupied, so we wanted to take as little space as possible. So what we need is a small sample. We use 15 microliters of plasma, so it's not much we need. Sometimes if you had an accident, you don't know if people hit the head, maybe it's the legs that's a problem. We don't want to use too much blood already from people injured, so we need to isolate the plasma and then put it directly on our electrode where we have immobilized the antibody if we find best. Then we introduce the sample, and if the GFAP is present, it will bind to our antibody, and then we use impedance measurements to kind of detect how the concentration is. We did a lot of calibration and did a lot of testing to see what the impedance was before adding a sample, and then after to kind of differentiate how much the impedance should be. And also we also saw a lot of difference between different people testing. So we also try to variate that people actually have different techniques as well within the testing. So, yes. This is about us, and yeah.
Thank you very much. I see there's a question.
Yes.
Could you please explain a little more your surface applications? What are
So we start with a gold electrode that's covered in gold. Then we use DBS as a binder to the antibody. And then we use BSA to block out the unbinding site to make sure that we don't have any unspecific binding. Then we add the sample and wash it away to only, because the plasma might contain something else as well, so we want to remove that again, and then we use ferrocyanide to measure it, because then we measure the difference. I call it the heartbeat of the, so you can kind of see the molecular level heartbeat because it will change the measurements.
Yeah. What we do is incubate for an optimized amount of time. So for each step, we have DSP to bind to our gold surface, then and has another binding extreme to bind with the antibody, and that is our setup. In order to not have unspecific binding, we then incubate BSA. But yeah, so what we did was test every step with impedance spectroscopy. The principle is that we first measure in a clean electrode. Then when we put the DSP, if something has bind, we only incubate in the working electrode. So you know that if something has happened in the working electrode, DSP binding to the surface or the antibody, we will have an increase of resistance. So we will see it in the impedance measurement.
We did multiple testing of each test, of each step, so we kept repeating to make sure that, because as I said, there might be some changes between the sun was out and when it wasn't out. So we would kind of do multiple testing on different days as well to see the range it would be in, and then compare them also so we could see both before and after. So we kept testing the different steps as well along the way, and every time we did an optimization, we would go back and do the same again to see how the optimization would change all of the steps.
Yeah, hi. I have a question on the build-up. So every sensor shown so far shows, okay, I wanted to detect it quickly, the GFAP, but do you happen to know how fast in the body the build-up of the GFAP actually is?
I believe once you hit your head, the GFAP increases within hours. So in 1 hour you will have already measurable concentration of GFAP in your blood. So not even hours, even less. If we can test very low concentrations, we could. Our test takes 15 minutes, so it's like a COVID test, maybe. So even half an hour, you can test for very low concentrations, and you already have a set amount of GFAP in your bloodstream. Then it can be tested. So.
Okay, thank you.
Yeah, but it could be relevant to do multiple measurements with maybe a time interval in between, because also when you hit your head, you don't always are aware what time you have hit your head. So it could be relevant to do multiple measurements.
What's for you the most creative aspect of your sensor?
We obviously chose this method because our lab also had experience with it, and we tried to. The thing is that it wasn't a successful studies, but what we are trying to also do is to try and reutilize the electrodes, or try to see how many measurements we can get out of each electrode, and there's multiple ways to try to chemically remove the binding of the GFAP, but that required a lot more time that we had. We tried to try and measure like how many times can we measure different samples, but so that since it's very tiny and very easy to use, that would be a good further up study in that sense. So
Yeah, so what we did was we wanted to see if we could measure multiple times on the same electrode, because one measurement might not be as precise. We had some variation, so we were in the middle of, we tried to see how much different there was between each test we did with the same electrode. We could see a bit of a tendency in the change, but we didn't have enough. We didn't have enough testing to see how much. But we are able to use the same electrodes with the same to test multiple times to give a better representations of the levels. But we need more testing for that to be completely precise. At the moment, the first one is the most precise we have. We're using PalmSense. It's a device we have available in our lab, and we saw that it worked well for our testing. And so we're using PalmSense.
So the accuracy depends more on how our biosensor actually works, not really the reader. So what we do is, we in order to raise the accuracy, we focused on getting a good calibration curve. What we use PalmSense in order to read our measurements and to have a software, because our idea also was not to give, not for the competition specifically, but it would be nice to maybe not give specific concentrations, but since we have a class system, use it like a COVID test. So you put your measurement in the software, and regarding the fitting of the resistance that you get, you can see if you have a severe, low, or medium, and that can also be done with like another software. But what we just used PalmSense in order to read our measurements, not to the accuracy of them. Sorry, I don't understand. No, we use pipetting in all of our steps, and because to make sure that we are not washing off in one electrode more than the other, we have like a specific time and way that we wash off the electrodes. But no, we try to do it as simple as possible. So it's just millicube water. During the functionalization steps, it takes more of washing, but the idea is that you would just put the sample, then wash it off with millicube water. If you want to dip it, it's fine. You just need to have it off and do the measurement. So no, it doesn't, no. No, so ferricyanide we use to do the measurements. To make ferricyanide, you have to use the specific powers for the chemicals, and you dilute them in millicube water. Thus, that's why, like, if you clean it in millicube water, you have a tiny, more diluted solution, but you don't need to air gun it or dry it out. Or that's fine.
The final question.
Well, I'm not exactly sure if I understand the question, but to my understanding, GFAP has like in the anti. Sorry, yeah, I wanted to say GSP, DSP. Sorry, DSP has a very it reacts with binds good with gold. Yes. Yes. Well, we followed what we saw in literature and also what our group had experience in, and DSP is also used for linking other antibodies or specific markers or specific binders in gold nanoparticles as well.
And we also chose this instead of, for example, EDC and SH because it's a one-step functionalization instead of having multiple steps that we would have to optimize.
Yeah, and also, sorry, we were testing at each step, and we were making sure that the time that we incubated the DSP was enough, was not too much and not too less, in order for temperature also to not affect it and to not. So.
Okay, thank you, detectives, for the pitch. Okay, the next team will be the team of Glasgow. Make sure you use the microphone.
Okay.
Good luck.
Oh.
Right. So, last presentation of this session. I think I can skip the bit where I say that TBI, very bad. If you hit your head, there's consequences. We're all aware of this. And I wanted to point out one important thing, is that on the market right now, there are biosensors for TBI. There was, I just literally was reading today, there's a biosensor from ISAT, I think, which goes for $3,000 approximately, and can detect samples from 20 microliters of plasma, 3 different analytes, so pretty good biosensor. But the problem is that the biggest amount of cases for mild TBI are in developing countries. So countries where the typical wage is a few dollars per day. So these biosensors can be quite expensive. Therefore, all our concept was focused on reducing the cost and trying to find an easy way to perform the assay. So the problem in developing a biosensor that is cheap and could be easy to use is that it might have cheap materials, so not very fancy technology applied to this biosensor, but this also means decreased sensitivity. Therefore, we focus all our efforts on trying to increase the sensitivity of the biosensor, but keeping it simple. Therefore, how can we improve the sensitivity? Well, we thought about scouting the literature. We found out that increasing the size and inducing threat could be a technique. So what we propose here is using a recombinant protein there's the drawing of our recombinant protein, that had 3 different domains. Well, clearly the GFAP domain, because this will allow the competitive binding to the antibodies fixed on the gold nanoparticles. Then HSA, so human serum albumin, because it's soluble, is well characterized, and as I said again, we are trying to make things simple. And we wanted to try and skip the blocking step. So calibrate the surface of our biosensor with this protein, by saturating it with this protein, might help us to skip the blocking step. And finally, superfold the GFP, because the emission spectra of this protein overlaps really well with the absorption spectra of 40 nanometers gold nanoparticles, which are the ones that we're going to use. This method will allow us to amplify the signal produced by our biosensor by substituting the big recombinant GFAP, let's call it FGFAP, with normal GFAP, which is almost 3 times smaller than our recombinant protein. So this is a bit a schematic of how the system would work. We first have the recombinant protein bound to the gold nanoparticles, and we have a shift in the absorbance spectra of the LSPR chip. Then, upon when we add the whole blood sample, GFAP would compete with this recombinant protein, and we would detect the blue shift. So the idea is that the overall shift is bigger than the shift just produced by GFAP. Another concept, so talking about threat that I mentioned before, is that we actually try to quench the fluorescence because we want the energy to be transferred from the fluorophore to the gold nanoparticles. And finally, in order to make things cheaper, simpler, and to use less components, we also try to avoid the use of a spectrophotometer, and we will actually wanted to just use LEDs of three colors, so that we will have different points set in the absorption spectra, and instead of detecting the shift in the absorption peak, we'll just detect the shift in the amplitude at these 3 points. Thank you for listening, and we are ready for the questions.
Thank you. I see there are some questions.
What about when you have non-specific binding of single proteins and those displace your FGFAP?
Okay, so the thing is that we picked human serum albumin because we're using human blood. So any other blocking solution will have this problem, pretty much. So I think that using human serum albumin would probably be the strongest blocking buffer for the blood sample. Also, many biosensors use plasma. We are aiming to use this with blood, because again, like, we wanted to make it simple. So I hope that answers the question.
You're directly using blood, right?
Yes, that is the aim, the ultimate, ideally. Well, yes. Ideally, the blood cells would cause like non-specific reading, but we aim to. So since the GFAP, the recombinant one, will already be bound to the antibodies, And the competitive interaction will have to be so specific that in order to have non-specific interactions, this could be very unlikely. So that is why we aim to use this system.
Yeah, thanks for the pitch. How about the performance of your biosensor? And does it work as good as you expected?
No, but I think that most of the biosensors do not really work as good as we expect. It's reality. I mean, trial and error is fundamental for development of any technology, I think. We did some calculation and some testing on how much the overall shift, so basically the big protein, FGFAP, has to bind to binds to these antibodies and produce a shift. This shift has to be at least twice bigger as the shift produced by normal GFAP in order for the amplification system to work. So the point is that in literature, we have seen that we use labels to amplify the signal. Our aim was not using labels. And when we use antibodies, the signal in some papers has been boosted of 300%, so like 3 times. And 3 times more will already be enough for us to detect a blue shift in this case, which is the result of the amplification. I hope that answered the question. Ideally, yes. Well, we tested it with actually doing a competitive assay. So we tested with plasma and then competition between the two. So as a competitive ELISA, but on LSPR, pretty much. We also tested it with in the way that we wanted to perform, but that didn't really give us promising results. But it's when we think about protein-antibody interaction, as Richard Feynman used to say, it's not like a lock. There is when something that has a lower KD and a higher affinity for the antibodies, it can actually displace the recombinant protein. So no, the answer is no, but yes. Because with the competition, we would have to not incubate, but we'd have to pre-mix the sample with the recombinant protein. In this case, it would be easy to just put the blood, wait for a bit, wash it, and then we hopefully get the result.
Yeah, so for detection purposes, we first took a sample like a design full wave spectrometer, which we already saved in there. And then we calculated the initial diode readings, like before the sample binding happens, and one after the assay happens. And then we compared those bindings to the values in the transmittance spectrum that we can see there. And from those amplitudes, we tried to estimate that if the shift had happened, where would have been the position. And we could do that by calculating the ratios and getting the local minima of the design wavelength. And from that, we were able to estimate the shift, and that's how we were doing. Yeah. No, so our setup was just 3 monochromatic LEDs and with a single photodiode, which was just taking in the amplitude of at the points of the dominant wavelengths of the LEDs.
So we use thiol binding, just the cysteines on the antibodies, just they are able to be passively bound to the gold nanoparticles just by incubation and then actually placing them on. Thiol is on, no, thiol is on the antibody. The thiol is found in the cysteine in the antibody, and that, because it has a slightly negative charge, can bind to the gold because the gold carries a slightly positive charge. Yes.
Anyone else?
My question may be a bit more curiosity or maybe a little understanding. So you have the HSA component of your fusion.
Sorry, I'm just reaching from the projector in this.
Yeah. So it seems like it's going to be a kinetics-based model of how quickly things dissociate or reassociate. Did you look at how HSA binds to other things? So I mean, basically in the blood, it's a carrier for lots of things. So if you're putting a blood sample on there, is it picking up other junk? And yes, it might be acting as a blocking agent for large-scale proteins, but it's going to bind to common drugs in the bloodstream, common small markers. Does that affect signal or anything like that?
Well, so basically, the question is, it could be beneficial or could be detrimental having HSA.
Mm-hmm.
So as I said, this is the ideal concept. So yes, it could, but at the same time, any other blocking solution, any other blocking buffer, blocking protein in particular, could have the same detrimental effect in this case. So
I was curious, I'm just not sure how far the testing went, so in terms of plasma or...
No, we tested it with plasma so far, because for the competition that was going to be used, we were going to be using plasma. But ideally, for a future commercialization, the blood sample would be perfect for an easy-to-use biosensor. So yes, this does require more testing, but in the literature, I haven't really found something that as the blocking buffer within the system itself.
Cool. Looks good. Thank you.
No problem.
One last question.
You mentioned you use these only 3 wavelengths for the detection.
Yes.
In order to make it faster, if I understand it correctly, or what is the idea behind that?
The main objective was to make it small as possible, and to make it cheaper.
Okay. And did you do any testing on repeatability? So you do some fitting to these measurement points. Did you see how reliable this fitting was?
So we had done multiple tests, and the answers were pretty reliable, but it was breaking down in some circumstances, like for example, if the shift was less than 1 nanometer, then it was having some trouble in detecting, but greater than 1 nanometer, we were getting good accuracies for that, and at a reliable rate as well.
Right. Okay, thanks.
Thank
you. Then thank you for the last page. This is the end of the first round of pitches. Thank you all for listening. Upstairs, there's the partner market, and you can also get a cup of coffee or tea. So, see you later. Hello everyone. Welcome. Hello? Yeah, okay. Welcome everyone at the second round of pitches. For the teams presenting today, make sure that when there is a question from the audience, the people in the live stream, they don't really hear that because they don't have a microphone. So repeat the question and then answer it. And also the other team members who might answer questions, they can sit here in front while you're presenting. I think that's it. So we can start with the first presentation. And that's from SensWork.
De klikken? Ja. En dit is gewoon de volgende, right? Test. Yeah? Okay, welcome everybody. My name is Janne, this is Marit, and we're from the SensWork team from Wageningen University, here today to give you our innovation pitch. And by the end you will also finally find out what the deal is with this €5 bill and our biosensor. But more on that later. First, we actually want to get started on the principle of our biosensor. So we actually make use of a quite well-established technique, the lateral flow assay, and many of you have had experience with this during the COVID-19 pandemic. And as you can see, it's made up of quite a few components, and how it works is that you basically put the sample on the sampling pad, and it will flow through the conjugate pad through the nitrocellulose, and this is just a special piece of paper. And then if the biomarker is present, a so-called sandwich will be formed, and this will yield a fluorescent signal. And the intensity of that signal is correlated to the severity of TBI. So that's how it works. So how did we actually get to the sensitivity we are at right now? And we actually worked really hard for that, and we've tried over 100 different variations of antibodies, materials, concentrations, buffers, and we actually started with selecting the best antibody pair, and we really tested all possible combinations, not just the recommended ones. And then we went into detail for the materials and the buffers, and we actually made use of theoretical calculations to determine the concentration of antibodies that we needed to spray onto our strips. So it is a well-established technique already, so you might wonder what's new. But it's actually more than you might think. So firstly, our test is actually quantitative, while actually all other lateral flow assays are mostly just qualitative. Also, we make use of europium nanoparticles, and this really yields a very distinct, interference-free signal. And it's also these europium nanoparticles that can be found in the €5 bill to check its validity. On top of that, we have a built-in sample preparation, so it's hands-free. We can measure in real time, and we have a specialized cartridge. And on top of that, we also have extra control lines added onto our strip.
Yeah, so we already mentioned that we did a lot of optimization steps to reach our biosensor. And it's good that we did this, because now we managed to reach a high sensitivity of up to 0.1 nanograms per milliliter in a detection time of under 50 minutes. Oh, sorry. With a detection time of under 50 minutes. So this is super fast. And also our biosensor is small enough to fit in your hand palm, as you can see here. And it's super easy to use for everyone and every user. But that's not all. Because lateral flow assay is a well-established technique, there are a lot of innovative features that could still be implemented. For example, there are mathematical tools that could shorten the detection time immensely, so you don't have to wait as long. And also multiplexing is very easy to do when using lateral flow assay. For example, you can use biomarkers in parallel to GFAB to make your biosensor even more accurate. An example of a biomarker that can do this is UCH-L1.
And to save our environment a little bit, we can also use biodegradable plastics, such as PO1, that can be printed with a 3D printer into our cartridges. And to make our biosensor even more accessible, which is the point of care, we can build an app. We can build an app that will allow everyone to see the data and process the data even more easily. So, in conclusion, our biosensor built upon the basic principles of lateral flow assay that makes it a rapid, accurate, and cost-effective point-of-care biotest that is accessible for everyone. And we, as students from SensUs from Wageningen University, overcame the first problems to make this test workable and innovative. And we hope others will continue upon our efforts in the future. But of course, this was not possible without the help of our sponsors and interviewees. So thank you. We're sensing it. Thank you very much.
Yes, thank you. Are there any questions?
Well, okay, the question is about the mathematical tools. So basically, it makes use of this, there is this kind of curve where like the concentration will really peak at a certain moment, but mathematically you can already know what the end product is going to be using some mathematical things like taking the second derivative, and then you can already, while the test is actually running, calculate what the end concentration is going to be, so you can make it even shorter. It's not necessarily extrapolation. I think the mathematical tools is like with the second derivative. So, well, I think you can call it extrapolation then. Yeah.
Other more questions?
Hi, thank you for a nice presentation. You mentioned on one of the slides that the test is hands-free, I think.
The sample preparation is
The sample
hands-free.
preparation is hands-free. What does that mean exactly? Like, how is it hands-free?
It means that you don't have to make use of like a centrifuge and pipettes and stuff like that to first put the sample onto our test. You can just put it on your test.
So it's still similar to a COVID test that most of us knows.
Yes,
in the
basically.
preparation. Okay.
But also there is actually more preparation. You have to do stuff with your nose and stuff. And you don't have to do that with this one.
Okay, yeah. Thanks.
Yes.
Okay, I think you maybe misunderstood us a little bit. The question was about quantitative and qualitative. So what we did was we turned a qualitative lateral flow assay test, which you use in COVID detection, and we turned it into a quantitative one using fluorescent signal. So that's because the COVID test actually uses gold nanoparticles, and then you see a line by eye, and we used fluorescent particles, which we could read out with a reader, our biosensor, which I showed you, and this makes it quantitative. Okay, good question. So the question was, how do we get a number, basically, from the fluorescent signal that we measure? So the reader actually measures the amount of fluorescent signal, and because we labeled our secondary antibodies into the sandwich assay, we can see how much GFAP is bound by the amount of fluorescent signal. So the more fluorescent signal we measure, the more biomarker is present. Does that answer your question?
You're welcome.
You have elaborated on, you've quite clearly communicated that the labeling is one of your most innovative aspects of your biosensor. Could you take us through how you got to this point? How did you select this type of label? How did you choose to use fluorescence?
Yes, of course. Well, of course, the entire goal of this competition was to make it quantitative. And there are a lot of, also optical markers you could use, but those are less sensitive in general. So fluorescence reaches a higher sensitivity, and because we needed to detect very low concentrations during this competition, we chose for a fluorescent marker. And because we chose especially for the quantum dots, those have even more optimal functions that could remove background, for example, or limit background at least, to make it more sensitive. So it was all about the sensitivity here.
Okay. The last question.
So yeah, thanks for the nice pitch. And yeah, making a lateral flow assay quantitative is a big challenge. Yeah, so I think it's out there in the field for long. So how did you find that? So the whole concept, did you read it in literature, or was it an idea out of your team, or was it coming
Do you mean
from...
like how lateral flow, the principle
No,
is founded?
the quantitative aspect of the lateral flow. Yeah,
So how that was found.
How is it your idea? Is it?
Well, we do work together with coaches, but of course we do, as students, have some input ourselves into the idea, and we actually discussed with the whole of our team, like, what do we want to achieve, what do we want to do. So also it's also coming from us a bit.
Okay.
So yeah.
Yeah, because it's also, you can translate it to many other assays.
Yes,
Of
you
course,
could also test, like, if you use other antibodies, you could test any other disease if you would like.
Okay, thank you.
Yes. Okay. Thank you for your pitch. And then it's time for the next team. Yes. Yeah, maybe one microphone works better. So okay.
Has it?
Yeah.
I'll click. You'll click, yeah. Hello everyone, my name is Max and this is Kirsty and we're SensUs. So GFAP in America is a really big problem, and we've seen that rehabilitation programs have costed even around $80,000 per patient. Our biosensor, being a point of care, low cost, quick turnaround solution to this, able to quantify GFAP, will help with this. The principle we are using for our biosensor is an organic electrochemical transistor, which is very similar to the extended gate field effect transistor. There's 3 nodes to a OECT, where voltage goes into the gate, and based on this value of the voltage, the channel current will change from the drain to the source. You can kind of see that characteristic channel current on that graph to the right, where it gets increasing, but eventually reach saturation over time. So we are functionalizing this gate in order to be able to detect the change in GFAP.
We functionalize the gate by adding a CNT film on top of the electrode.
the CNT film has carboxylic functional groups attached to it, and the antibodies then bind to those carboxylic groups, which then bind the GFAP, which changes the gate potential. We tested our technical feasibility on multiple processes, first of which was the CNT film. We were able to develop a film that can produce up to 90 electrodes per film, and we also verified that the organic semiconductor material, which is the P.PSS PEG-DE solution, was able to remain on the electrode while in solution, as well as the CNT film. This is very important, especially since we traveled from America to come here, so we were able to verify that the electrodes would still be functional after traveling all this way. In the middle, you can see our electrode, which the blue dot is the channel, which is the P.PSS solution again, the gate, which is the CNT film, and then the source and the drain. On the right, we were able to measure GFAP by measuring known concentrations of GFAP and looking at a specific time point and applying these to the modified Hill equation. Or fitting with the modified Hill equation. We faced some challenges during this development and researched literature to help us find some solutions. One of which was P-dot remaining on the electrode while in solution. Since it is water soluble, we added PEG-DE to allow it to remain on the electrode in solution. And we also were able to find that vacuum filtration was the best mode of creating our CNT films so that they were stable while on the electrode.
More into how we prototyped our system, we developed it to be a mobile, point of care, very quick turnaround, a low-cost GFAP biosensor. So as Kirsty was describing, we started with our electrode, which was the OECT, and then based on how much GFAP binded to the gate, its channel current changed. From there, we found the channel current to be very small. We're talking nanoamps to microamps. So we needed a very precise amplification board. And we custom made a circuit, printed a PCB, and the main source of amplification involved a transimpedance amplifier. From there, we needed a way to read this voltage that was being amplified. It was going from current to voltage. So we used the Analog Discovery 2, which is a multi-purpose electronic device that it can measure the voltage as well as write voltage at the same time. And the really nice thing about it is that we were directly able to program it within MATLAB, which, as you can see in the top right, we made a custom user interface within MATLAB that's able to gather this voltage while running the measurements. And you can change parameters such as the upper bound, the sampling rate, and the duration. Finally, 2 proprietary devices in our prototype. We had a microcontroller, which in the future is going to enable wireless communication, like an app on your phone. And then we had a power board, which allowed 1 micro USB connection, and everything on our board was powered. Thank you for listening to our presentation. We'd be happy to answer any questions you guys have.
Thank you for your presentation.
Very good talk. So this type of devices, usually polymer-based and P.PSS-based devices, there is this channel drifting problem, which I face in my research. So what is the solution for this? What do you think when you work with the real sample?
So the question was, I believe, how do we ensure that the P-dot remains on the sample while it's in solution?
Even though they are on the surface and they stay there, there's still this we call it channel drifting, but the channel potential, channel current is changing all the time with different concentration of analyte, even though you have the same base solution. This is
it's a very small amount of P-dot that we place on the electrode, and we are ensuring that we have repeatability in the process, and then we're also using PEG-DE to make sure that the P-dot remains on the electrode, so that we do a bake of the electrode, and through the baking we're making sure that the P-dot is uniform.
you do some processing to
Yes.
to keep
Mhm.
it stable. Yeah.
Correct.
That's good.
Yes.
Thank you.
Mhm. Oh, and sorry, to further elaborate, we also, we have a machine called the Biodot where we are able to precisely place very, very small amounts of the P dot in the exact location. So that also helps with the processing.
So, yeah, thanks a lot for this presentation, and I think it's also quite a nice concept. So it's rarely published in literature. Yeah, so there are only few teams working on such concept. What I found really novel was using the carbon nanotubes on the gold electrodes. So there are, thousands of ways to functionalize a gold electrode. Why choosing the carbon nanotubes?
The carbon nanotubes really increases the surface area, which is allowing us to get a better potential and stability of the electrode itself. Yeah.
Yeah, so we have used self-assembled monolayers for gold electrodes in the past, but we have a lot of issues with variability using those. And the CNTs are just shelf-stable in comparison to self-assembled monolayers. So that was our approach there.
Yes, it's more or less the answer of for the last question. Yeah? So it also stabilized the open circuit potential of the electrode.
Yes.
Yeah, that's a really good question. So essentially what we did is the time before the testing event when they gave us the different concentrations for the samples, we used our electrodes and we ran it through our systems and we found the characteristic curves again. And what's kind of interesting is that we were noticing the voltage was dropping a lot quicker, so I think the transportation might have had an influence on that. So we were able to just change our numbers in our code, recalibrate it, and worked good.
So I did not touch on that. That was more to elaborate that we tested 2 different antibodies while we were developing our electrode, yes. So we ended up using the high test.
Are there any more questions? Okay, yeah.
So we still have time, right? So then I put another question. We don't release you earlier. So you mentioned in the electronic circuit, it's only nanoamperes you measure. So typically the P.PSS transistors, they have high currents. So why is the current so low?
That's a good question. Like we noticed when
he's not wrong.
when we were doing it like with just the P.PSS, we were noticing it was already in the nanoamps range. So we were like, we're gonna make this super overkill board. We're gonna get this transimpedance amplifier going into this second stage where it's buffered, where we have this dual buffered digital potentiometer that's making this very precise analog voltage, canceling out the offset from the first stage. And then there's a whole second stage where we're using a summing junction. So I'm more of the electrical hardware guy. I don't explain the chemistry as well, but if you guys need a really good amplifier, here I am.
Okay. When do you fly back? I need you.
Marketing.
The marketing, good. So the P.PSS, this functionalization or this mixture, maybe there's some issue with it. So I don't know. Yeah. So we use P.PSS, and not such stabilized matrix. Maybe there's an issue, but I don't know.
if anyone has still one question, yes. Then the last question for you.
Did you also measure in your calibration curve that you show on this slide on the bottom right any GFAP concentrations below 2 nanogram per ml?
Below 2 nanograms?
Yes.
Is that the question? Yes. We measured down to 0.1, I believe.
This is in picogram, yeah, but in thousands of picograms. That's why I was asking.
Oh, yes. The IP switched from nanograms to picograms, the information package. So we had displayed this in picograms, but we measured at 0 picograms, and then to 0.1 nanograms, which is the lower range of the TBI.
Yes. Thank you for your pitch. Yes, the next team is Pils.
Right. Hi everyone and welcome jury. My name is Lander de Waele and today I proudly introduce to you EASY, our user-friendly, fully integrated biosensor for GFAP, the biomarker of traumatic brain injury. Easy because performing a measurement with our device is, as its name suggests, very easy. All you have to do is scan the QR code of your package, insert the cartridge into the device, add your sample and activate it with a simple press. After that, the device will do the rest, and once a measurement has been performed, your results are shown on a graphical user interface and can even be stored in a protected database, allowing easy access for authorized doctors. Now, you might wonder, how does this all work? Well, let me guide you through the different steps. First, we need a sensitive bioassay to capture GFAP out of a plasma sample. Now, to increase the sensitivity, we expanded the surface of our electrodes by adding gold nanoparticles functionalized with antibodies on top of it. And to ensure specificity, we block the surface of our gold nanoparticles with BSA and that of our electrodes with superblock. To make the step to a point-of-care device, we integrated these electrodes into our innovative iSimple cartridge. And this can be seen as a lab-on-chip device that allows for fully autonomous sample processing based on paper microfluidics. Now, how does it work? Of course, first you need to add your sample to the inlet. After that, with a simple press, you will activate a pump which will pull your sample into the cartridge and activates a timer. Once your sample has been pulled into your cartridge, it flows through a microfluidic channel and is incubated on top of the electrode. And by making use of a so-called plug timer, which can be seen as an hourglass, your incubation time can be tuned. So, as you can see, once the liquid in that timer moves from the left side of the channel to the right side, your incubation time is over, and a redox buffer will be pushed over the electrode, washing your sample away. Now, the last part of that redox buffer will be again incubated on top of the electrode, and by making use of an infrared detector, our device knows exactly when to start performing a measurement. And this, of course, makes our device fully autonomously user-independent, which is a huge advantage. Now, how do we measure? We make use of an electrochemical impedance spectroscopy, or EIS. And this is a sensitive way to detect changes in the resistance of your functionalized surface. So by adding more layers to your surface, the resistance will increase. And by making use of a QR code, our device knows the baseline measurement, so before sample addition. And after sample incubation, we do a second measurement on field. And the larger the relative difference between those two, the more GFAP is bound to the surface. Finally, our results are shown on an intuitive user interface, integrated in a compact design, and can send to a protected database, which can be accessed by doctors via our developed Pulse for Doctors website, and even via API calls. And this makes, of course, that our easy package does not only include a sensitive and fully autonomous biosensor, but also a complete and intuitive readout platform. Thank you for your attention, and we're willing to answer any burning questions.
Yes, thank you very much. I see there's a question.
20 nanometers.
So regarding the sensitivity, we still need a lot of optimization because we encountered a lot of problem during this summer. So we switched to this because it was a protocol that we know it's working, but we didn't reach the sensitivity, but we know the technique that we would use if we would, because there is also a lot of literature that support this. Regarding the specificity, we still try like the difference with just plasma and GFAB. So, and it was there was no difference with plasma and an increase of GFAB. So we know that we are still binding and measuring something. I think I could an exercise. Yeah. So the different data is because we do. So basically, we do the difference between when we add the plasma GFAB or only plasma against the last step. So it was a super block. And you can see that we definitely see a bigger increase with the GFAB when there is 10,000 picogram in it compared to just plasma.
Yes, again.
We don't think so because we did read a lot of papers about using this kind of technique and
So, again, similar question I asked the teams in the first pitches. So, if you would summarize in 2-3 sentences the most creative and most novel part, because over the years we have seen similar devices from Löwen. So, what did you create really novel in your device now? Great job. So thank you. Great job.
Yeah.
Please note to repeat the question from the public.
Well, of course, your bumps are just paper, and there is like a bit of variation in between them. But what we want to achieve, because this timer is highly adaptable, you can make it longer, shorter, and what we actually want is to perform a measurement when there is equilibrium. So then it doesn't matter if you are 2 seconds or 3 seconds too early. It's not like in minutes or something. That's absolutely not the case.
So basically, because we were a bit time-wise struggling, we could not go very innovative regarding that. So we just use a protocol that works by adsorption. So we basically bind the antibody on the gold nanoparticle by adsorption with a methodology that we know it's working. And we checked, for example, if the protocol was working by putting a luminescent antibody on top. It was working really well. So I think we have also. Yeah, for example, so that was a nanoparticle without a fluorescent antibody. Then we just put the same concentration antibody that we would to functionalize the nanoparticle, but in a droplet. That's just the pure antibody in the middle, and then to the right you directly see. So basically there is the ring effect. So because it's a droplet, you see the antibody there at the edges of the droplet. Yeah, so yeah. But we knew that we would have a problem with droplets, so we also created a sort of cell system to functionalize the electrode because we didn't want all the nanoparticle to go on the side, and so we basically created ourselves a sort of holding liquid for every functionalization step, so we would have something equal that would just sink in and not get this droplet effect either. But that's not the electrode. That was just like a glass. So that was just to show the functionalization work. But then when they want to bind on the electrode, the ring effect will not apply because of the cell system.
Thank you for a very nice pitch. Could you elaborate on the choice of impedance implemented in this kind of flow system? Why did you choose that over other techniques?
Well, we try to find a way to do it label-free because that's just most time efficient in the chips we can make. Otherwise, we need extra chambers. We need kind of different storage as well. And we came across EIS, and that's a very sensitive way to measure differences in the resistance of your surface. We also try to do cyclic voltammograms, but based on what we read in literature, EIS is just more sensitive than a cyclic voltammogram. But yeah, it also has disadvantages like your electrodes are not always the same. That's the reason why we incorporated a QR code to compensate for them. So.
Did you consider optical techniques as well, or you just settled on electrical?
An optical technique is definitely possible, but then our device will need to scale a bit up, of course, because now our device is really portable, and we even got a developer board from PalmSense. So if we just have the chip of PalmSense, then it will be even smaller. So yeah, that's the reason why we choose electrochemical.
All right, thanks.
You're welcome.
Then it's time for the last question.
Thank you. This is going off the last question on EIS. I might have missed it, but you had the typical EIS plots and you were looking at charge transfer resistance. But I didn't see in the microfluidic system, are you introducing a mediator into the system, or how are you getting charge transfer if it's label free?
So let's go to chip. So what you actually have is in this chamber we store like a redox buffer with ferrocyanide, ferricyanide, and that, yeah, it gives like the.
So you could use the
electrons
same
you need.
chamber for a label system, right?
Well, it's not really a label. Well, yeah, you can see it as a label, but if you want to do something that also needs like a buffer on top of it, then you need an extra chamber like here with antibodies, and then an extra one with a buffer solution. So, yeah.
Thank you.
Yes, thank you again for your pitch. Yes, the next team is Ike Sense. Make sure to repeat the questions from the audience.
Okay, so as you may know at this point, the topic of this year was traumatic brain injury, or for short TBI. And to tackle the problem and detect TBI, we developed GaugeIt, an electrochemical-based biosensor. It makes use of ion-sensitive field effect transistor technology, or for short ISFET, but with a twist. The twist in our case is that we use indium tin oxide, or ITO, as channel material for our biosensor. Now, you may have heard of this material in applications such as touchscreens, or like airplanes, or solar panels. But in all these cases, it's used as conductive film. For our application, we need it to behave like a semiconductor. And through literature research, we found that it's actually possible to tune its properties and to make it an effective semiconductor for our biosensor. And that's what we did. We designed experiments to tailor the oxygen flow rate during the sputtering deposition of the material, which is what alter its conductivity properties, until we reach the optimal condition for our use. This allowed us to obtain a cheap, easy to manufacture compared to like graphene and other semiconductors, which is also transparent and custom tailored. Together with the ISFET technology then, this leads to an innovative combination for these type of biosensors. And the working principle of the ISFET is quite simple from this diagram here. We have source and drain made of metals, then the semiconductive ITO between them, and then we have an insulating substrate underneath. The ITO is functionalized with antibodies, so that when the samples arrive, the GFAP, which is the biomarker of interest for TBI, will bind to the antibodies. Since this protein is naturally negatively charged, it will then attract positive ions present in the solution. And due to the proximity with the ITO surface, this will in turn affect the impedance between source and drain. And we can then connect the ISFET to a potential stat, for example, and read out this impedance signal. This is a photo here of one of the chip on the cartridge that we manufactured ourselves. We all did everything from scratch in clean room, both the chips, the cartridge, and also functionalization and characterization. You can see here it has 4 main sensing areas, and each one of them consists of 6 ISFETs. These 4 areas approach will allow us to go also for multiplexing in the future. We also designed a microfluidic PDMS-based cartridge that will then let the sample flow through these sensing areas. This on the right here, it's what we used, the final device design we used for this competition. It just needs like a potentiostat down here, then some cables and electronics, and then the cartridge chip carrier here. Also, the use is quite straightforward. We just need to deposit the sample in the PDMS cartridge here, and then wait for 1 minute for the reaction and plug in the chip. And after 2 minutes, we can read the results and know if we have TBI. But our creativity didn't end here. We also designed what our first prototype could look like, an all-in-one prototype. You can see here, thanks to the low components needed, we can miniaturize most of them as well, also as the chip, and obtain a compact and portable all-in-one device, which is not bigger than our mascot here. And so to summarize, Gauge It, our idea starts from using ITO as a channel material, then merging it with ESFET technology, which is established, effective, can be miniaturized, and doesn't need any dilution of the sample. And overall, the device that results from this will be compact, portable, also easy to use, and can be quickly prototyped, as I showed in previous slides. So thank you for your attention. If you have any question, I'll be pleased to answer.
Yes, thank you for your pitch.
Okay, thanks for the question. So the question was regarding the GFAP protein. It's negatively charged, but there are other proteins in the plasma which are also like charged. And so how do we deal with these like type of unspecific signals, I guess?
So we have immobilized 5 different antibodies that are specific to GFAP. So we're expecting specific binding because of that. And we also used bovine serum albumin, or BSA for short, for blocking to make sure that we wouldn't have non-specific binding. But because we have 4 different wells on our chip and we could leave one unfunctionalized, we could have a differential read. So basically we can immediately compare sample with it on a surface that's non-functionalized and sample on our surface, which is functionalized for GFAP specifically, and see what the difference would be there in the readout. Oh, I'm sorry. Did you want to speak?
Yeah, no. So the question now was, sorry, can you say again? Yes, sorry. So for our competition here, actually we couldn't use the one I showed you because we had problems with that, like last minute problems. So instead we just used like 4 open wells and we directly pipette on the sensitive area there. But the idea for like later stage of this microfluidic is to have like sample which is whole blood and a filter so that it can filter out the plasma from the whole blood. And then we have like one inlet, and then these 2 channels which will make the plasma go through the 4 sensing areas, or even more if we want, and then out to the final collection area.
From your presentation, I gathered that the use of ITO was quite critical for you, but I did not catch why you wanted to use ITO and not, for example, silicon as a semiconductor.
So that is actually a good question, because like the advantages that the reason we went for ITO, for example, is, okay, compared to graphene, we can agree it's like cheaper and easier to manufacture, but silicon is also a good candidate as a semiconductor. For our specific technology, like making these chips, these ESFETs, and scaling up from that, we believe that using ITO, which just literally requires 1-step sputtering process to deposit, will be more convenient than using silicon, which needs more machineries, more steps. And so that will lead to an advantage in that sense. And another advantage is that ITO is also transparent. So in the future, we could also benchmark this with more conventional optical-based platforms, or yeah, basically that. I don't know if I missed out something or.
Does anybody has a question?
I have more of a curiosity. So I had the similar question about ITO, but using these types of devices, ISFETs and other devices, can scale down in physical size of the individual device. How does that affect performance? Does it improve decay performance, or is that something you looked at in terms of?
So to scale down the actual size of the ISFET, for example.
Yeah, or scale up. How does?
Yeah, so basically, if I will try to answer that, if I get something wrong, they will correct me. But the main point is that we can definitely scale down, and in the future we will try to go for that, so we can have very small chips and also reduce the cost of the materials. And it's probably one of the problems of scaling down is that the channel area will also reduce. But we use, I don't know if you've seen it back there, but basically we had a comb structure for the ISFET, which increased the channel area. So even if we scale down, we can use the same structure to then have a like higher area for the channel, which increase also the performance of the device.
Is that okay?
Yeah. I studied.
I saw that you used silanes to immobilize the antibodies on your surface, and these are typically somewhat liable to hydrolysis. So have you considered the shelf life of your sensor as well?
In the scope of the competition, we were not able to test a shelf life longer than 2 weeks, if I'm not mistaken. Right, but we definitely want to do that in the future. We're using silane as the primary surface where we would have free amines, so we can bind the antibodies on top using EDC NHS, which is a well-established protocol, and we did not have any issues while using the device and testing it at home lab in Aachen, with hydrolysis.
Do you have any strategies to improve stability if it would turn out to be a problem, or any alternative ways of coating the surface?
We did not need to test for that, but I'm sure this is something we would love to research further. So we're sure that our shelf life is then months and even years, maybe.
If I can just quickly add, like our goal ultimately would be to do more of this testing and reach for something that has a life of 8 to 12 months at least in the fridge. But I've seen it's possible, like there's different products also from Siemens and other companies that last for even longer. So definitely there's something there. We will need to find the best combination.
Good, thanks.
What is your mascot?
Yes. I made it, I crocheted it. Our mascot is Stella, an astrocyte. That's where the name comes from as well. And astrocytes are the glial cells in the brain and the central nervous system in general that create GFAP. It's a very important protein in them, and it's the one we were looking for. So we did not crochet a neuron, we crocheted an astrocyte. That's the important cell here.
There's time for still one more question. Okay. Then,
if there are no more questions, then I can quickly say that with the chip that we already have, we could, using other antibodies, immediately multiplex. And because we have 4 wells, looking at S100, for example, and UCHL1, we could just multiplex exactly what we have with the protocol that we're using currently. So we did not do it for this competition because only GFAP was in our sample, but we could do it in a week.
Okay, thank you.
Thank you.
The next team will be Make Sensing. This is the next slide and this is the previous slide. And make sure to speak into the microphone while presenting.
Yeah, and I go present to just do this.
Yes, then we'll go to the next slide.
Okay. Good afternoon. We are Make Sensing, and today I will present the design, the technical concept, and the approach of our TBI sensor. Our project is an electrochemical biosensor designed to give accurate prediction on milder to severe cases of brain trauma. For that matter, we divided our team into two, one responsible for the transducing system and another for the biochemical assay. Now, I would like to show you our ideal model. This is a ELFA with sandwich structure in the working electrode using anti-GFAP to detect GFAP through the signal of the electron flux. We built the electrode using laser-induced graphene, and they were linked to the antibodies using deposition of gold nanoparticles through cyclic voltammetry, and it was verified by SEM. To identify surface changes in the posterior antigen binding, EIS was used. To obtain a stable and sensitive signal, our sensor is a 3-electrode system with 3 key components: the antibodies, the potentiostate, and the gold nanoparticles. These components help in the sensitivity and the functionalization of our system, where the gold nanoparticles help bind anti-GFAP to close to graphene. Anti-GFAP selectively binds to GFAP, that's our analyte, and the potentiostat performs and reads the EIS and the cyclic voltammetry measurements. This implies a working electrode that's coated with anti-GFAP, a reference electrode that provides a stable reference point for the potential of the working electrode, and a counter electrode that is responsible for the flow of the electrons. Once GFAP antigen is bind, changes on the surface result on a potential difference between the working and the counter electrodes, and this allows a quantitative test since the resistance increases in a proportional fashion to the concentration of the target analyte. And now I'm going to show you the graphics. Here we have imaginary versus real impedance graphs, where we can see that with the surface changes, the resistance increases as the curves become steeper. We are able to distinguish between modification steps once we see that the results are similar when performing the same step. To increase the limit of detection, we started to test conjugates of gold nanoparticles and antibodies and orthoradic peroxidase proteins. Sadly, it was not functional enough to bring to the competition. So that's our future perspective. However, this system could still be measured using cyclic voltammetry and has the plus side of being widely used in the biosensing applications. Thank you for your attention. We are Make Sensing.
Thank you very much for your pitch. Are there any questions?
Yeah, it's dihydroxide peroxide, but yeah, sorry. Oh, all right. Yeah, these are hydroxide peroxide that was there. But we have Sure. All right. So, yeah. Okay. Yeah. In imaged, we have hydroxide peroxide that will be used by peroxidase to reduce and produce electrons. But what you didn't understand? You just didn't see that? No, no, no. It don't make damage in the antibodies. I'm sure about that.
Other questions, maybe?
We actually are sure about the graphene are produced because we saw about that results with Tem and Sam to verify if we are really producing that. So we don't have the results here because we don't have the time to show all the results, but we are sure that.
I saw a very nice visual in the beginning where you showed this 3D rotating lateral flow assay, I'd say. So what's the relationship between this and the electrochemical detection that you are doing later? Were you planning to use an electrochemical readout for a device that looks like this?
Yeah, actually, it's not the final model because we are missing the rest of the sensor here, the electrode sensor, because well, it's our first model and we didn't have time for the rest of that. But actually, we wanted to measure the analyte with the cycle voltammetry with the reaction that we mentioned in the last slide.
So we should envision a lateral flow assay where the lines are electrodes, and where you are doing electrochemical detection on both of the lines.
Here, not both lines. On the test line, it will be the work electrode. The control line it will be only in the colorimetric line to ensure that we have the antibodies. So, yeah, the conjugated antibodies was there to ensure that it's a control line. So it's a simple control line like the COVID test, that one. Only the test line will be the work electrode.
But you would use the sensor without an electrochemical control line? So without...
No, we don't want that because we want to quantitative measure. Without the electoral control line, we can't do that quantitative method, and this will be just a qualitative method like the COVID test normal that we have. There is a LFA, not a ELFA.
Then how would you account for non-specifically adsorbed proteins from plasma?
Non-specific?
Non-specifically adsorbed proteins from the plasma. How would you correct for that?
We have a pad here that will be a filter, like a filter that will not allow some proteins pass.
But it's impossible to have a filter that prevents all the serum proteins from binding on the
not all
chip.
the serum, but the bigger than the GFAP. And after that, we have the antibodies that are blocked with BSA2, so we don't have any specific binding, and the antibodies will bind only in our analyte. I think so. It's
It
like
seems
ideal idea, so it's theoretical.
It seems a bit optimistic, but okay.
Any other questions? Okay. Then I want to say thank you. Yes, the next theme will be toekomstsense. Make sure to really speak into microphone.
Okay.
And the questions from the public, please repeat.
Yes, all right.
We are Team Toucan-Sense. Welcome to our creativity pitch. Our biosensor, the Specialist, uses a custom-designed screen-printed electrode. The first layer consists of silver, followed by different coatings. The reference electrode is printed using silver-silver chloride ink. Both working and counter-electrode are printed using platinum ink, allowing for a quick preparation without surface activation. The blue layer serves as a protective coating. On this electrode, a GFAP-specific antibody is immobilized using L-cysteine as the linker. When the sample is applied, GFAP binds to this antibody and any remaining sample is washed away. To ensure comparability between electrodes, a reference impedance measurement is conducted by introducing a redox solution. Enhanced sensitivity is achieved by incubating with a secondary antibody labeled with gold nanoparticles, followed by another impedance measurement. However, measuring a single biomarker is not sufficient for a reliable diagnosis of TBI. We therefore plan to expand our method to multiplexing, which would enable the simultaneous determination of 4 biomarkers. In our initial approach, we employ 4 distinct electrodes, each with its own molecular recognition unit. We've already designed a microfluidic system and a cartridge to ensure precise sample preparation and prevent cross-contamination. In our second approach, we consolidate the multiplexing setup into a single electrode, minimizing resource requirements and sample volumes.
In order to provide a measurement process, we have designed and produced our own cartridge with a reading instrument and a user interface. The user interface provides step-by-step instructions on what actions to take. Furthermore, it displays the measured temperature within the cartridge and presents the concentration of the GFAP at the end of the measurement. To prevent medical staff from spending important working time, the user is supported by microfluidics, adapted to the measurement. Each solution has its own input channel to the measurement chamber where the electrode is located. This is easy to use, has a higher accuracy, and allows controllable flow of the solutions. Moreover, the previously mentioned multiplexing capability is possible. Not only does the results shows on the display of our cartridge, but also on our self-programmed app. With the app, measurements can be easily assigned to specific electrodes by scanning QR codes. And it simplifies the workflow of the staff. In this manner, they do not have to be around the biosensor throughout the measurement, leaving more time for other tasks. It also enables the direct entry to the digital patient's record, and notifications for responsible staff like medical staff. This is a specialist, and together we can sense the future.
Yes, thank you for your presentation. Do we have any questions?
Yeah, so sorry for the voice. So the question was which other biomolecules we're measuring with the multiplexing. And it could be the interleukin 6, which is also correlated with TBIs, which was the target in the last year's SensUs competitions. And there's also one protein, the S100B, which also is made or produced by the astrocytes, and it's secreted in the blood when you receive a TBI. So there are more biomarkers also correlated with the TBI. Yeah. Yeah, for the competition, we were like strictly only measuring GFAP. Yeah, so in the middle, it's like a bit grayer. It's the reference electrode. And then the outer one, like the circle one, is the counter electrode. And then you have the 4 distinct working electrodes with the different molecular recognition units.
With those 4 separate working electrodes, how would you envision functionalizing them for the separate analytes, since they're so close together?
Yeah. Yeah, the question was like, how do you immobilize the different molecular recognition units? And that's quite a problem. So we already designed a cartridge for the ones for the first iteration you can see here. And we have like rings inside, so there are no cross-contaminations. And this will be a big task to complete. And we're thinking about a setup like this one here, but we still have to design and develop. So we're just first at our first approach with the vision to the second one.
Yeah, thanks. Maybe I missed it. Does the sensor work?
So yes, we tested it, and we had a good calibration curve when we came here. In the really testing event, we had some troubles, some inconvenience, but we measured, and we were waiting for the results today in the evening. So yeah.
Okay, maybe in the following question, I mean, in your optimization, I mean, you only targeted multiplexing or also kind of sensitivity? Do you have ideas on how to improve the sensitivity, or are you saying that the sensitivity is good enough? I don't care, I'll just go for multiplexing.
So our first step would be the multiplexing, but still, of course, the optimization of the sensitivity is a big cause for a biosensor, right? So there would be also some development in there. But now our first approach would be the multiplexing.
Okay, thank you.
How did you deal with non-specific binding?
So we linked our molecular recognition to our electrode, and then as most of the teams did, we blocked with BSA to prohibit non-specific binding. And as we also said in the presentation, we're doing a reference measurement after the GFAP incubation. So if there is some cross reactions or so, we have it with our ref measurement, and then we're just comparing the ref to the second antibody measurement. So there shouldn't be that much cross reactions. So that's how we try to eliminate those errors.
So thank you again also for you. So where's the creativity? So what is really novel, what you guys developed, and can you describe it in 2-3 sentences?
So first of all, because of the whole system, so we increase the handling of the biosensor. So as we said, we have the whole production of every product, the cartridge, the SPEs, and the microfluidics in-house. So we printed everything ourselves. This is the first advantage we have, an innovative thing. And the second one is the handling aspect. You don't have to forget, because we designed this app, and it's supported by the microfluidics. So it's possible for also not teached staff to work with this electrodes, and it's spending very much working time for the responsible personnel.
Good, thank you.
So, yeah, no problem. It's very good user interface. I think you thought very deeply through this. So my question is about this previous slide that you showed, the microfluidics, and then here. So you are also using the redox probe, right? So how it's really working here? So one of the chamber reserved for the redox probe, and then when you mix it, activate it, and come to the surface.
Yes, so one of the chamber is for the redox solution, and you have like pumps attached to those reservoirs in the future, and then when we're ready for the redox solution, it would be brought into the reaction chamber on top of the electrode.
Currently, all the measurement that you have done, you just mix your sample with the redox probe and measure, right?
No, we first, so one of the reservoirs is washing solution, so we would wash off our sample and then apply the redox solution.
Yeah. Very good.
Yes.
To prevent cross-contamination between the measurements, we wash the whole channels before we measure with puffer and different solutions. So we tried to decrease this fact. In special, we doesn't make extra reference measurements just for the microfluidic chip, but we does it for the electrodes.
Are there other questions, maybe? Okay. Then I want to say thank you. Make sure to speak into the mic.
Okay, I will try. Hello everyone, I'm Emir, and hello everyone, I'm Emir, and I'm gonna present our creativity pitch today as on behalf of Team Biomarkers from Istanbul Technical University. Okay, this is actually a plasmon-based biosensor, and we are using this principle in our sensor. In plasmon-based biosensors, there's always on the surface like nanoparticles, and then the top of the nanoparticles, antibodies are binding to this surface. And then when the sample is dropped to the sensor, a change, like it could be a resonance change, and in this case it would be a SPR system, surface plasmon resonance system, and it could be an absorbance shift or absorbance change. So in these sensors, we sense the change that is provided by the nanoparticles actually on the surface. The advantages of the nanoplasmonic biosensors are improved sensitivity because the nanoparticles are providing the sensitivity that is required for the samples. Also, label-free detection. Since we are using nanoparticles, we don't have any secondary antibodies for the systems. High throughput screening, affordable and easily fabrication, and adaptable to point-of-care systems. This is our system, actually. We are using gold nanoparticles at 60 nanometer, and we are using spherical ones because it is more feasible and more optimum for our system. We are coating the surface with gold nanoparticles with the electrostatic interactions. We are using a polymer that is called as PLL, polyl lysine, and since the gold nanoparticles are negatively charged and PLL is positively charged, the electrostatic interaction is achieved by them. And on the top of the gold nanoparticles, we connect the antibodies with linker, which is called PEG. So the PEG is also providing the preventing the unspecific bindings and providing a little bit more sensitivity. And when the sample is dropped, GFAP is bound to the antibodies, and we observe a absorbance shift. In this system, we are using a absorbance change because it gives us more sensitivity, actually. And yeah, actually, we adapt this system onto a 96 well plates, so we could use for a lot of people because we are living in crowded city and crowded country. So when a crash, a car accident happened or a bus accident happened, a lot of people can go to the hospitals, and with our system we can sense the GFAP and we can detect the TBI for 96 people at the same time. So this is why we are using the 96 well plates. Actually, we did our plate reader by ourselves from scratch. It is actually a basic spectrophotometer, but we wanted to do it from ourselves because we want to make it more affordable and more cheaper. So yeah, this is our results. We did the optimization of the PLL, which is called polyl lysine. So we used 500 microgram per milliliter for PLL, and for the gold nanoparticles we are using 200 microliters because it is the optimum, because they can coat the surface with equally actually on the surface, so it gives us an optimum results. And we did our validation with glycerol before. So when the glycerol concentration is increased in the sample, our system is detect this increasing with absorbance changing. So it is a validation for us. Thank you for listening to me. I can have your questions for now.
Yes, thanks a lot for your pitch. And it's the last time for questions. Are there any questions?
Could you explain what is the mesh layer that you showed underneath the gold layers on which you immobilize your nanoparticles?
Oh, with PLL actually, like it's a polymer, and we're using this polymer for coating the gold nanomarkers on the surface.
Ah, right. And the PED linker that you use to immobilize the antibodies on the gold nanoparticles.
Yes.
What are those? What is it? What are the functional groups and what is the material of the linker?
Did you want to?
One more time.
What is the PED linker?
PEG actually.
Oh.
We bioconjugate our antibodies with PEG so that we can functionalize our gold nanoparticles with the antibodies, and it also prevents non-specific binding. Instead of using BSA or any other serums, we could also use PEG because it also helps antibodies to I don't know how to explain, but push it up a bit so that it prevents this non-specific binding.
So this PEG has a thiol group and an NHS ester?
Yeah.
Okay, can I hear it again?
Do I understand correctly that this PEG then has a thiol and an NHS ester?
Yeah. It's with it connects to the antibodies with thiol binding. So yeah. We did the actually binding in outside, like we mixed the PEG and antibodies outside for preventing the unbinding, unspecific binding. Then we dropped the PEG and antibodies together to our system. Thank you.
Other questions, maybe?
You explained that you made a really custom device. Did I understand correctly you build a custom spectrometer as well?
What?
Did I understand correctly that you also build a custom spectrometer?
Yeah, we did our spectrophotometer by ourselves, like for decreasing the cost actually. The other spectrophotometers are really sold with high prices, so we try to do it with less cost, so make it more affordable for hospitals, for public hospitals, and the other health services.
Actually, we were looking for the 500 nanometers between 700 nanometers. So, for 1 nanometer shift.
When the GFAP is bound to the antibodies, there is a 1 nanometer shift in absorbance. So we try to sense the 1 nanometer shift with our spectrophotometer. It's actually a basic spectrophotometer with less cost, actually. Thank you.
Yes, so you mentioned a shift of 1 nanometer at what concentration of GFAP?
You are asking for limit of detection, or?
Yes, so or the underlying question is how quantitative can you be?
Okay, for now, we just achieved 100 microgram per milliliter, but in the further, we are trying to decrease this limit of detection with the new additions maybe, and that.
So, but 100 microgram per milliliter, that's very high, or?
Yeah, it is very high for now. We didn't do it for the time that we have given, so yeah.
And then the other question, what are your ideas on how do you deliver the sample to the well?
What?
How do you deliver the sample?
What?
How do you deliver the sample to the well?
I didn't really understand that word.
We can drop the sample by ourselves with the micropipette.
So
For the time being.
and then you would do that 96 times to have the entire busload of patients, or?
We didn't. I don't know how to explain it for the time being, but we didn't purchase a device, or we didn't design any automated system for that for the time being, because we had a limited time, and our antibody is also delivered so lately. So we were trying to optimize the system with PLL and PEG antibodies. So.
Okay, thank you.
You're welcome.
Are there any more questions?
Sorry, can you like volume up so I can hear? Oh, okay. What was the purpose of the usage of the glycerol, right? Just we have to do a validation for our system, but our antibodies and proteins are delivered us very lately. So we just want to validate our system with glycerol, since we have no protein and no antibodies in our labs, and we are waited for so long for the shipment problems. So yeah. No, for testing the system, like did the gold nanoparticles and the other stuffs are sensible for the concentration changing or not? It is the validation of it. Like, yeah, it is like that. It is not a specific binding. The glycerol binds to the gold nanoparticles. I don't know the exact principle of it, but with the concentration change, our system, the absorbance shift is also changing. So we want to do validation for it, since we have no antibodies and no proteins. Yeah, we are looking for absorption actually. Yeah, we didn't do anything. What? Particles is present there. So particles are providing us the absorbance. Like the shift is actually done by the gold nanoparticles, as we see from now on. Yeah. Oh, it is not. Yeah. Yeah, it is not like SPR, because SPR is looking for the surface change, surface resonance change. We are looking for the absorbance change.
Okay. Is there anybody with still a question? Okay, then I want to say thank you.
Thank you.
And this was also the last presentation. So this is the end of the second round of pitches. I want to thank the audience for your attention, and I also want to thank the jury for today. And at 4:15, the intermediate program will start in the same room as the opening was. So I see you there.